So, welcome back to this course functional genomics, and this is the second lecture of week 1. So, in the previous lecture we looked into how the field of molecular biology has evolved, and that led to the emergence of a new era called as Genomics Era, right. So, that is what being summarized in this slide here. We see that we started with the Mendel's concept to the DNA double helix, how Watson Crick solved the structure of the DNA, and then all the codons, and then you have for the first time developed what is called the recombinant DNA technology in 1972, and that led to what is called as the DNA sequencing, right, by Maxim and Gilbert in 1977, where you have now a tool by which you are able to sequence the DNA. So, the moment this technique was developed and shared with all the scientists, and you will find that there are so many labs now they are able to make the recombinant DNA either from the DNA itself or from the RNA by converting it to a cDNA or copied DNA, and they started sequencing. So, obviously, you are going to have large amount of data coming up, because we know that the DNA sequence is made up of 4 bases, the combination of which gives the sequence. So, that led to a new concept called gene bank, meaning it is a bank which stores not currency, but the sequences. In 1982, the American establishment went to form a database called gene bank database, where if you sequence a DNA, you are able to deposit the sequence, and anybody who is trying to understand the function of the gene or DNA can also access this database. It is a free open database, and that was the first one, but soon there was another such database called EMBA from Europe, and Japan also developed very similar kind of a sequence database. And this revolution has led to understanding of the genome better, and then the first gene, or for a human disorder, the gene was mapped using purely based on sequence that are present in the chromosome. These are repeat sequences they have used. 1983, a gene was mapped, and then soon they have understood the power of understanding the genome that would really help us to understand how the diseases are caused and how the different races of the human population are different and gives them unique advantage. So, then the American government came up with a kind of a concept that we should sequence the human genome using the technologies that were available, and this was still a proposal, but that was in 1984. Then 85 the PCR came, because that made things much easier. Now you do not need to really wait for a large amount of DNA, you can have small amount, you can make millions of copies. And then likewise the technology, it is not only the chemical based approaches, but electronics and instrumentation based approaches also evolved. Then there were discoveries or inventions which led to what is called as automated DNA sequencers, where you can sequence a large number of samples. A computer is able to analyze the sequence and give you the sequence straight away. That came in. So, that really led to what is called as an explosion, because then people started using this methodology for sequencing the human DNA, and 1987 the first generation human genetic map was developed, a kind of what is called as markers, which are landmarks, kind of a milepost in the genome. And then they came up with that concept, and then 1988 the Human Genome Organization was formed, and US is National Research Council of the US issued a report that we should map and sequence the human genome, and then as a prelude the East chromosome was sequenced, and that is how they started testing the methodology and the challenges that come along with analyzing the sequence and how to store them and so on. So, this has led to a new era that you call as genomics, the birth of genomics. And 1989, the first a successful discovery of a gene causing a human disease without knowing what is the function of the gene. This is called as positional cloning. That led to the discovery of the gene for a disease called cystic fibrosis. So that's the first one. But soon that approach, that is, you are going to use certain markers, these are nothing but repeat sequences, and use them to understand, identify, locate a gene has become much easier, and thousands of human genetic disorders genes have been identified since 1989. The cystic fibrosis gene was identified. So, going to this lecture we are going to run through some of those important mileposts. So, you can see that with every advancement, when you talk about human genome sequence and so on, it also comes with certain issues. The issues could be, when you sequence the DNA, and then when you want to put that sequence available in a portal, in a database that is accessible for everyone. So, that is one of the major decisions taken by the Human Genome Organization, that all the sequence should be deposited in a portal that is accessible to anyone and everyone. So, that also comes with its own challenges, like if you have the sequence of different races, and if somebody identifies what are the sequence that gives you a risk of developing a disease and so on, can this be used against certain races or individual. So, there were ethical, legal and social implications that were discussed, and certain guidelines have been formed by the consortium. And then these discoveries, understanding the sequence and coming up with markers led to, as I said, many new genes that are implicated in disease process, one of them being the breast cancer gene, what is called as the BRCA1 was mapped likewise. So, again it is a very landmark discovery in terms of cancer genetics perspective. Then with the such advancement, we have several landmarks. Then in 1992, the second generation human genetic map was developed, which clearly tells you where are the markers for every chromosomes. The density has gone up. So, people are able to use these markers to understand and investigate the chromosomes better. And then you have finally the human genome program, sequencing program was established. It is a 5-year plan. It was established, and then there were consortium mode, because there are about 5-6 countries came together, and they have kind of understood and agreed upon as to which region of the genome they should sequence. And the Wellcome Trust Sanger Center joined the force in speeding up the process of sequencing. And in 1994, the human genetic mapping, pretty much the goal was achieved. They are able to make pieces of the DNA of the human genome, label them and sort them, and able to put all the mileposts. So, you have pretty much the skeletal framework has been done for the human genome sequencing. So, that really hastened the process of sequencing. Complete physical map, the distance between different segments of the gene and others came up in 1995. And then with all this advancement, the bacterial genome, which is much smaller, the influenza genome was sequenced. And then there were other issues that came up along with certain, when they have sequenced the genome, and then also US government came up with a policy how genetic discrimination in workplace cannot be tolerated. So, again these are ethical issues, right. The other simpler organism like yeast sequence was also completed in 1996, because these are much smaller genome. And by then the scientific community also sort of understood that just sequencing the human genome alone will not suffice. We need to have very good models to understand the gene functions. So, one of the models that are closer to the human yet very good in terms of genetic tool was mouse, and therefore they decided that even the mouse genome should be sequenced. And then genetic map was achieved in 1996, and then there was the consortium which together discussed, and it is called as Bermuda Principles, that really agreed upon that any such large scale sequence initiative funded by the government, be it the US or the Europe or the Japanese or Chinese, the data should be in a public domain, therefore all can access. It is called as open data policy. That was agreed upon in 1996. So, that really has changed the way people can look at the genome sequence, because these centers have understood creating the data, that is generating the data is one, but analyzing them and understanding is going to be much more Herculean task, and therefore it should be open. Therefore, many groups who have competence in analyzing the data can do. It is going to really help the society. So, that is the reason why they went for open source data being deposited. Then came the genomics era, 1997 to what you see now. There are large number of genome sequence are made available. So, you have, for example, many initiatives that came, for example, E. coli genome, one of the common bacterium that is present in your gut, E. coli was sequenced, and then there are other centers, for example, which started sequencing other models. For example, the C. elegans genome sequence was initiated, and they also developed other ways. For example, it is not only the sequence that helps us, we need to understand what is the variation between two individuals, or the one that are healthy and not healthy. If really the DNA sequence gives you that attribute, then even the variation should be understood. So, there was another initiative which really helped to understand the single nucleotide polymorphism, or the variations that came in. And then in 1990, that is when really the full scale human sequencing began. People started doing it in a rapid pace, because technology is developed. You have very good automation came in, instrumentation came in, they are able to do better. And then 1999, of course, the first human chromosome, that is chromosome 22, smaller chromosome sequence was released. So, that led to a series of data release with, for example, Drosophila genome was sequenced, mustard, another plant sequence was released, and then 2001-2 you see that human draft version of the human genome sequence was published, and then mouse sequence was published. And then also we started in parallel sequencing all the RNAs from different tissues. You take tissue from the human body, convert the RNA into cDNA and sequence, therefore you know what are the genes that are expressed in a given tissue. That also began, called as mammalian gene collection. So, which led to the sequencing of human cDNAs. So, that is how it went on, and 2003, of course, the finished version of the human genome sequence was complete, pretty much what we have now for the human genome. Whatever region of the chromosome could be sequenced was completed in 2003. We have them in the portal. So, anybody who wish to look into what are the genes present in a given chromosome, you can go and look, and there will be a session after a couple of classes that we would introduce you to various such databases. So, you can also learn how to browse through this database and then understand how the genes are. So, as I told, it is the sequence information that we have now, so many different organisms being sequenced and so on. It was an enormous task which did not come only from the lab, genetics or biochemistry based approaches. The way the science was done, the genetics or molecular biology was done, has changed from a lab centric to more industry like approach. What I am showing here is a photograph from Morgan's lab, where one of his co-workers studying Drosophila genetics. All you see that at the back rack, which is the bottles are kept, are different strains of bacteria, Drosophila being maintained there. That is how it began, the genetics began. But in 2000 or so, when molecular biology was really at its prime, this is how the labs looked like. So, you have lab benches, we have certain equipment, but still it was a manual work. So, you have to take the solutions, put them in a tube, allow the reaction to get over, and load it on a gel, analyze, and so on. Still, it required the manpower to conduct experiments. But things have changed when, with the advent of what is called as genomics. It was only possible because of the automation and a large number of biomedical equipment that were developed in parallel to carry out many of these experiments without human intervention and to analyze the data. The what is called as high throughput screens all became possible in the later 2000 onwards, and it has become like, if you look into large genomics labs, it doesn't look like the lab that was shown in the previous photograph. It is more like a kind of an industry, and this is called as industrial scale because you scale up everything. There will be hundreds of people working, there are hundreds of equipments, thousands of samples being sequenced at the same time, and it led to data that is being generated just like anything. So if you look into the equipments, there is a revolution, if it's not just evolution. Most of you must have used microscope in your college or school. That's something that is shown on the left side, a simple microscope that was used during Morgan period. But that's no longer, you cannot use that for when you are analyzing large number of cells. So now you have what is shown on the right side is a workstation, what you call as. You can pretty much put the sample and program it. The microscope would look at the cells, score them, pattern, and give you the data. So that's without anyone sitting there. That is possible because the technology has developed, and that kind of an approach is required if you want to look into the dynamics of large number of samples. One person cannot really do all these things, so only machines can do. So that technology really played a major role in changing the way biology done, be it looking at the cell, even in liquid handling, for example. The one shown on the left side is some of you must have used the pipette that in colleges you must have used for taking the reagent, to what you call as calibrated little handheld pipettes, to now you have workstation that is shown on the right side, with the machine which you give a program as to what volume of sample should be added in which tube, and it does in no time and with precision without any error. So that's how things have changed. So now you can pretty much set the system wherein you have, for example, cells put in tubes that are grown there, and you can ask the system to extract the RNA, convert that into cDNA, and sequence it, analyze, and give you data. So everything can be now automated, the platform would do, and at the end of the day you are going to have the sequence which you have to validate, analyze, and so on. So that is how the system has changed, and that led to more challenges we will talk about a little later. So likewise, when you talk about sequencing, which is one of the major contribution in the genomic field, that the sequence of the DNA or RNA being generated. So the one shown on the left top is an electrophoretic setup that developed, that's how some of us have done DNA sequencing in 1980. So these are the manual gels. We have to run it, and then at the end of the day you have to expose the gel to X-ray film, and then you will have bands. Then you read the sequence. In a day, probably 100, 150 bases you can do, and this has changed with the advent of what is called as automated sequencers in early 2000. So you have machines which can take up to 380 samples, do all the sequence on its own, give you the sequence in soft copy, be it in a server or pen drive, whatever it is, and you can analyze them. So without any human intervention. So things have changed. So there were companies really helped in developing such technology. One of them was Lloyd Smith, who introduced the first automated DNA sequencers, and that was done by a company called Applied Biosystem. We really changed the way the human genome was sequenced, and that led to the scientific community to take up many different organisms for sequencing. Now even these approaches are outdated. We will discuss about the contemporary approach people use for sequencing the DNA. In this period, generally the molecular biology, genetics, this was pretty much done by research labs which are more interested in understanding discovery based science, understanding how the genome functions and so on. It was not a commercial entity, it was like a pure science, but the implications of understanding the genome into the health has become even a business, because everyone wants to live better, live longer, disease free. So all of us go to doctors and they prescribe medicines. The medicines come from pharmaceutical industries, which are not non-profit organizations. They are for profit. They do R&D to come up with drugs. So the genome and its variation and its implication in the health has also changed the way industries look into this field called genomics, because now they have interest, and the molecular biology has gone into industry. That's when you came up with an industry called biotech industry, which uses the genome knowledge to develop new therapy or therapeutic protocols or intervention strategy or drugs. So that's what's something shown here in this slide. 1990, the Human Genome Project was launched. 96, the Bermuda Principle that is drafted for Human Genome Project, saying that the data generated is free for everyone. Since it is free for everyone, there are also people who are interested in understanding the data and using it for as a commercial point of view in terms of therapy or drug and so on. In 98, one of the famous researcher launched a commercial company called Cellular Genomics, and they said that we will finish the human genome faster than the Six Nation Consortium, and they went with altogether different approach for sequencing the DNA, and they have used heavy usage of computers, we will talk about a little later. And finally, they gone into many other discipline which could be of commercial applications. 99, the chromosome 22, first human chromosome was decoded, model systems, genomes have come. 2001, first draft of the human genome was released, and 2003 Human Genome Project was completed. When this was announced, it was not only the Human Genome Project, that consortium, but even the Cellular Genomics by then, within a span of 4 or 5 years, they have completed the human genome sequence as well, so which the other consortium took several years to complete. So these are some of the famous captions. One, in 2003, you can see that Time magazine has shown two people. One is the Francis Collin on the right side with specs. He is the one who was championing the Human Genome Consortium project, and on the left side is Craig Venter. He is the one who founded the Cellular Genomics and completed the genome sequencing part much quicker than the other did. The reason being, they have used a very different approach, and they have used what is called as computational tool to analyze the sequence, which really helped them to finish faster. This is also the time when things have changed. What you see on the left side is a setup which is like industry like, thousands of people working and cutting down the cost by using a large number of equipments and computational tools. And that really made a huge difference. And this gentleman, Craig Venter, who founded the Cellular Genomics, not only did that, but he also established a new institute called the Institute for Genome Research to understand how the genome and variation can contribute to health and disease, and then the Craig Venter Institute and so on. So he was also the one who made a synthetic living organism by putting together DNA sequence of different organisms and made a new species, of course, which are lab centric, which never come to the free atmosphere. So now he is heading this a company called Human Longevity, where he is trying to make the living much better without much disease and so on. That is his ultimate aim. But he is known for also the speedy discovery of gene. When the human genome sequence was started, the aim was to sequence the genome, and people have used various approaches to find where the genes are. So, it is a very laborious and challenging task, but what he went and did was to do a different kind of approach called EST, which stands for expression sequence tags. The approach is that you make cDNA libraries from various tissues, for example human body, and then sequence only a part of the cDNA. Therefore, you have about 100 bases randomly here and there for a region that is transcribed. Now, you go back and look at the genome sequence and see which region has identical sequence. That would tell you where are the genes are. So, that is an approach that was developed by Craig Venter and really helped in understanding where the genes are located. Otherwise, it was extremely difficult because, as you know, less than 1% of the genome is represented in transcript. So, it is going to be a Herculean task to find where the genes are. In that way, it was revolutionary in terms of vision and so on. And these approaches also led to enormous data, the wealth. If for example, if you look into the human genome sequence, if you pull out the DNA from our body, it can go all the way to moon and come back. That is the length of the DNA. So, and if you get the sequence out of it and from all the organisms, how are you going to really store the data? How are you going to analyze? It's humanly not possible. So, that led to a new field of research and methodology development called bioinformatics. So, it is also primarily now we talk about bioinformatics as something which is a computational analysis of genomic data. It could be DNA, it could be RNA and so on. So, it is a field which has a specialization that falls between three major domains. Of course, one is biology because these are biological data. Then you have computer science because in the data science what you call large data analysis come from approaches by which you are able to automate the process the computers can take, analyze and give you data. Since you are analyzing the biology data, it is called as bioinformatics, but it also involves a lot of statistics, mathematical modeling and so on. That is where it is in the cross section of biology, statistics and computer science, and that is being called as bioinformatics. So, is it that bioinformatics came much later than genomics? So, most of us, majority, even the common public hear about bioinformatics. You must have seen there are various institutes and universities even offer bachelor's, master's degree in bioinformatics. There are bioinformatics papers introduced as well in other curriculum. We talk about all these things only after the Human Genome Project has been launched, because the wealth of data that was generated, there could be a large number of labs that could really look into and understand. So, the need or the demand also has gone up, just like the information technology boomed in the country and other part of the world. But it is not that in late 90s or 2000 the bioinformatics came in. It is there in existence much before. In fact, the very term bioinformatics was coined by Ogeweg and Hesper in 1970, but the intention was not necessarily to analyze the genome data only. It was like the study of informatic processes in biotic systems. That is their definition. Any information that comes from the bio field needs to be analyzed using certain automated processes, because it could demand such analysis. It is not humanly possible. That is how it was proposed, but not very well practiced by this team. But you can look at it is not only in 90s it came in much before. I am just putting some time scale here. It all started probably with the development of what is called the Atlas of Protein Sequence in early 1965 or so. So, that is when people started analyzing the protein. If you remember, I said after Morgan, his students started working on protein, and protein became main tool to understand how possibly the cell functions. Biochemistry field evolved, and then they started sequencing by cutting the protein into smaller peptide and then cleaving them into individual amino acids and so on. So, when they did the process, you need to, how do you arrive at the sequence? They have used certain simpler algorithms, kind of, machine reading tools, and to come up with the sequence. So, that is the kind of approach that was taken in early 1965 using, computational approach, which pretty much being used now in peptide sequencing since then onwards. As I said, there are in 1970s there are algorithms, computational tools came in to understand the protein and its structure and so on. And then with this, there are large number of proteins being sequenced, like we talk about gene bank, which came much later. There was a protein data bank which was developed, and all the protein sequences were deposited over there. So that, so in that way, that is also a bioinformatics, in the sense that they are storing data from biological origin for analysis. So that obviously bioinformatics existed even before the DNA sequence came in. And then there are many other softwares came in until 1980s to understand the protein, and 84, 85, that is when the DNA sequence came in, because the sequencing technology or the tool approach was established with Gilbert. And then of course you have 1982 the gene bank and so on, the first phage, lambda phage genome was sequenced and so on. So like from there you have wealth of data that is more of genomic, whether it is from the Drosophila or a human and so on. You will find that this genomic data overtook the protein sequence, and no longer people are sequencing the protein, because the moment you have sequenced the DNA or the messenger RNA via cDNA, you are able to predict what is a coding sequence, and if you are able to predict what is the coding sequence, with no time you can predict what is the protein sequence. So, the protein data bank, after which all derived from the sequence of the mRNA, meaning cDNA. So, now unless for identifying what protein it is, like for example 2D gel electrophoresis, people really do not go for protein sequence, because they just sequence a bit to know what is the sequence and go back and look into the protein sequence derived from the mRNA and match this is the protein. So, the protein sequence is pretty much now shelved, people do not do. But this came up with much more challenges, because the volume of data that comes from genome sequencing data have really grown. So, there were new technologies, but many of them are based on the earlier principles as to how to analyze the DNA sequence. But with the advent of RNA sequence coming up, expression analysis coming up, and the variation projects, the demand for newer technologies and newer approach to understand the difference have come in. So, that is what you will see little later. But you cannot say that the field of bioinformatics, meaning classically putting a database wherein information regarding biological systems are stored, if you define strictly that way, is not that new, not even 1960. Taxonomy, if you talk about classification of organisms, which a subject people hate, is very important, because that is the way you are able to relate organisms, classify them. You know that India, for example, has these many plant species or animal species, very important. And that is nothing but a database, wherein you have put in the data as to what are the plants exist, what are the different species, what is the characteristic species, what is the relatedness. This is again a database which can go back in centuries, you can trace back. But not only that, even it started with domestication of the animals, domestication of the plants. If you go back, even our own ancient literatures, whether it is Ayurveda or whatever, it talks about medicinal plants with certain name and within certain characteristic feature. These are the plants with this property available in this part of the country, can heal this process. Again, it is a database. So, it is bioinformatics is not something new. It exists as long as the human civilization came into place. There is a process wherein the data is stored, retrieved, ever since the language evolved you have. And there are even before that, there are it's as old as human civilization itself. So, so if you relate that, but what has become important in recent years is the development of computers and computing tools that really changed the way the data being analyzed. Earlier it was like stored and analyzed by humans, but now the data is so voluminous that humans cannot really analyze the sequence. So, that is again you see that there are some timelines given here. 1951, the first commercial computer came in, that is around when the peptide sequence being sequenced and analyzed. So, obviously they have used that tool. IBM came up with storage device, because this is important, because you need to store data, because the data size increases. The removable storage drive really helped, because you can put that, fill it and take it another, put one, and that really helped us to store the data for later analysis. 1962 and 81 came the workstations, the workhorse behind most of the data analysis applications, Apple and Sun Microsystem. And in 1983 the Apple personal computers came, and 1990 is when the internet or web servers came and has changed the way the data being analyzed. So, anybody sitting from any part of the world can analyze the sequence using remote login. So, that was possible because of the web servers. Just to relate with the contemporary media tool that you guys use, 95 is when the famous browser, Internet Explorer, was released. 2000 Wikipedia, which is a free source for most of the information. 2004 is the Facebook, and 2005 is YouTube. So, all these use data that are stored in different part of the world, and to query, analyze whatever you want. So, it is a very similar tool that being used for even bioinformatics, but much more complex data and complex tools to study them. So, the bioinformatics, mainly the one that is involving the genomic information, has changed the way science was done. So, there are new thoughts that came in with the birth of the post-genomic bioinformatics field. So, earlier people used to use what is called as a hypothesis-driven science. Now, with a data science, you can do hypothesis-free science. I will talk about what is the major difference between these two. And data mining as a prototype approach to hypothesis, this data really helped to start what is called as a open exploratory science, without really thinking that you have a model, you want to test the model, that is a hypothesis. And collecting and classifying, that is something that is normally done in classic mode of science. And two, understanding the molecular mechanism, for example, understanding how the gene expresses and so on. This could not have been done without the bioinformatics field, because the challenge is much more. That is something we are going to discuss. Let us see what it is. So, the hypothesis driven research is something that is shown on the left side. Okay? So, you have what is called as a theory. So, you have a theory saying that humans evolved from monkeys. This is a theory based on observations, because we have a pair of hands which we use for purpose other than walking. Monkeys do the same, and you have a pair of legs that are dedicated for walking, monkeys do. So, we can say that, but we have some abilities that are beyond what monkeys can do. So, we would say that we are more advanced, more recent as compared to monkeys. Therefore, humans evolved from monkeys. This is an hypothesis, right. So, then you strengthen hypothesis, you do few more tests, and then you confirm that possibly that is true. You cannot prove it, but you can say that is most likely. So, the way you can do is you can go back and look at human genome and monkey genome, and take, for example, a dog genome or a snake genome, and look at how similar or how different we are, and you find that the monkeys are more closer to your genome sequence than, say, the dog or a snake, then likely that we evolved from monkey. This is the way to prove that. That is called hypothesis driven research. Now, the data science with all the bioinformatic tools, it has changed. So, what you have is huge amount of data. So, have one file in which you have the human, the other file you have, for example, monkey, the other file you have frog, you have fish genome, and so on. I do not do anything, I have no hypothesis. All I do is I am going to compare all these 5 different files in terms of how similar the sequence are. I do this and then my patterning, we basically look at certain patterns and then it is going to tell you there are more patterns that are similar between human and monkey as compared to say a monkey and dog, a monkey and snake and monkey and frog, whereas frog has more similar pattern with snake as compared to human or dog. So, this kind of patterns led to an hypothesis. Now, we can say that dogs evolved, the frogs evolved earlier than snakes and dogs and monkeys and humans. So, you can come up with a theory now. So, this is hypothesis free science. That is possible because you have a data which you can mine and then look at, but these two are not really independent of each other. The hypothesis driven research, that is conventional science, provides more models to test even using data science, and the new theory that is generated from the hypothesis free science using data science can give an hypothesis which again can be tested again using the conventional approach. So, these are not something not overlapping, they are in fact complementing each other, but there are various ways of doing research. So, if you look into the entire discussion so far we had from Mendel to the sequence bioinformatics, how does it really, that is the comparison. So, Mendel's hybridization experiments led to theories of inheritance, dominance and segregation, and then we came up with the chromosomes and linkage, wherein we say they are the distinct entity, some genes are present on the same chromosome, and then of course, the chromosome nothing but DNA, and you have they replicate, make copies, and you have gene sequences, and the genome is sequenced and you analyze and so on. So, this is how you sort of understand the evolution. So, really how does it, all these different steps or stages of understanding can be categorized. So, this is how we call now. So, Mendel's hybridization experiments and principles you call Mendelian genetics. The chromosomes behavior during cell division, either mitosis, meiosis, classically called as cytogenetics. When you look at DNA molecules, manipulate them, change them, it is molecular genetics. When you sequence the entire genome, when you go for a large high throughput sequencing, large number of data if you are generating, it is normally called as genomics, and the large data you generated, we use computational tools to understand. So, that is called as bioinformatics. So, these are man-made distinctions, just to say that each subdomain requires different skill sets to perform and to understand and to master. So, that is what it is, but it is all same assembly line. In fact, even today people do tests like Mendel did. All the high yielding varieties of many of the paddy, rice or wheat that you find are all based on classic breeding experiments. So, it helps you even today. It is not that Mendel did and we are done and forgotten. And we understand how different these varieties are, what gives that a particular advantage to particular variety, that it gives you more yield. We can go at the molecular level and then understand. So, that comes molecular genetics or genomics or bioinformatics. So, it's everything is integrated, so you cannot leave them, but this is not the end of the road. So, with every new approach, every new tool and our understanding of the genome, we can go and look at something beyond what people have done, because we have seen that what you talk about molecular genetics was not known to Mendel or Morgan. They did not know what DNA is, but we now know. So, the next how this particular information led to emergence of a new field is something that we are going to discuss in the next class.